Well, hello and welcome everybody. My name is Gloria Isabel Giraldo Calderon, and I will be one of your instructors today. But before we get started, let me introduce you to Dr. Marianne McDowell from the University of Notre Dame, who is one of the 3 co-PIs of the ViewPath project. Thanks for being with us today, Marianne.
Thank you, Gloria, and good morning, or hello, everyone. It's not morning for everyone. As Gloria mentioned, I'm one of the joint PIs of ViewPath DB, of which VectorBase is a part of. And some of you may have already known that I took over as a principal investigator from Frank Collins in the previous iteration of VectorBase. So welcome to the third in a series of 4 webinars on how to use the new resources in VectorBase. We see that some of you are repeats and some of you are new. So I apologize to our return guests, but as what I'm about to say will be a repeat for you, so you can just tune out, I guess, for this part. But for those of you that are new and didn't already know, VectorBase has been part of the Bioinformatics Resource Center program, which is funded by the National Institute of Allergy and Infectious Disease within the NIH. The program originally started in 2004 with 8 different resource centers. And the funding occurs in 5-year contracts. And over the process of the last about 20 years, they have reduced the number of resource centers, originally from 8 down to 4, and now down to 2. And so what that meant is that VectorBase merged with UPathDB, and UPathDB supported bioinformatics resources for eukaryotic pathogens and fungi. And so now we are one, and we're ViewPathDB. So we started merging in September of 2019, and we're now complete, which means that the legacy VectorBase, the old VectorBase, will go away in a couple of weeks. And so now we're excited that you're learning about the new resources and tools that we're providing with ViewPathDB, and today's is on transcriptomics. We realize that change can be difficult, but we feel that this merger is providing a lot of new tools for the vector community, and the outreach staff is excellent, and I'm sure that they're going to give you a good webinar today. So thank you for joining us. I'll turn it back over to Gloria.
Thank you, Maria. Now some logistics. The webinar will last for about 1 hour or less, and this is the outline that we have for today. The main goal is to answer your question. Our main topic is transcriptomics, as Marianne said, and we will demo, different aspects of the website with examples to showcase what we have. Omar and Marianne, are you able to see the second slide? Last time they
froze. Yes, it's moving. Yeah, perfect.
Perfect. Thank you. Okay, so not only during this webinar, but always the outreach team is ready to answer your questions and hear your feedback using all these communication channels. The email link in the contact us, the page chat, social media pages, Facebook and Twitter. This is an image of how the screen looks to you as an attendee. And at any time during the presentation, please type your question in this box. It is private, only we can read it. And the outreach team in the background will be happy to answer them. As Omar posted already in the chat, we are recording this webinar, and the video will be up in our YouTube channel. Sorry. So we have a playlist in our YouTube that's all the webinars, and this is how you can access them. You go to the homepage, click here in YouTube, look for the playlist, and look for the webinars. And if you want to look at all the webinars what we have recorded in the last year, or even just the 4 of this webinar series, you can go to help, learn how to use ViewPath, click on webinars, and here is all the list if you don't want to go directly to YouTube. And as you notice here, this is a link showing you the steps that I just highlighted here. As described in the invitations we sent out, this is an introductory webinar series. We will use VectorBase as examples, but because the layout of all the ViewPath sites is the same, you can do anything that we are doing to demo with any other of the parasites or fungi sites, using different data sets, but the same layout, tools, and resources. And if you scroll to the bottom of the page, and here I'm showing you with this arrow, these are the links to all the other sites. And here, for example, the first link is the one, the main ViewPath site. And if you click here in this mosquito here, you will get to the VectorBase website. The icons to the right of the footer, meaning these three here, are also related to the ViewPath project, but the layout is a little bit different, and you can find also dedicated videos from our playlist to know how to use them. And here I have highlighted OrthoMCL, which you can use to investigate gene homolog relationships for all the genes in the parasite, fungi, and vector-based websites. CleanEpi for the exploration and analysis of epidemiological studies, and microbiome, which is a data mining platform for interrogating microbiome experiments. Today, the instructors are Omar Harp from the University of Pennsylvania, and I, Gloria Isabel, from the University of Notre Dame, who will both demonstrate how to explore transcriptomic data in VectorBase, and we are going to cover these major points. How to use the searches, and the previous webinar was very in detail, all the background information about the searches, but today Omar is going to focus just on how to use them for transcriptomic purposes, and I will show you how to explore the gene page and the genome browser. One last thing before I move it over to Omar is that the strategies he is going to construct step by step with you are just a guide, and the types of strategies or number of steps you can add depends on your research question and your creativity. But if you want to, get an example, just go to the website, click on My Strategies and Public, and we have 20 examples for you. Omar, now it's all to you.
Okay, thank you, Gloria. I'll go ahead and make myself the presenter. Okay, so hopefully you can see the VectorBase homepage. Okay, yes. Thanks. Okay, perfect, thank you. Okay, before I get into the transcriptomic searches, one quick thing to note is this banner at the top here. It occurred to me that we should mention something about this. So you'll notice on when you come to VectorBase, there are a number of banners that appear on your page. We have this cool tool where you can collapse them and hide them, but you can always show them again by clicking on this little speaker icon. There is one announcement that I would like to point your attention to, and that is or NIAID, as Marianne mentioned, NIAID is one of our main funding agencies, is conducting an impact survey for the BRCs, so the bioinformatic resource centers like ViewPath.db, which VectorBase is a part of. The survey takes about 5 minutes to take, and one of us will put it as well in the chat, so you can click on it there, or if you come to the VectorBase homepage, you can click on it, and we would appreciate if you take those 5 minutes and answer the survey and let NIAID know the impact of the BRC has had on your research. Okay, so as Gloria mentioned, I'm going to walk you through some transcriptomic searches. And when we talk about transcriptomics, obviously we're talking about gene expression at the transcript level. And the assays that we include, there are a number of assays, obviously, that can explore transcriptomic data. And these include things like if you run a PCR in the lab on your cDNA, for example, you're essentially probing the transcriptome of a specific gene. But even things like express sequence tags, which are right now and considered an old technology, but those also represent a transcriptome. I'm not going to be talking about ESDs today, so I will focus on newer technologies like microarray and obviously RNA sequence data. So in VectorBase, you can explore specific genes or the genome browser, which Gloria will tell you about. But one of the strengths of VectorBase and all ViewPath.db databases is the ability to explore all the data, the underlying data, in a way that allows you to ask your own questions and develop your own hypotheses. And I will start doing this right now to demonstrate what it looks like. And today we're going to be looking at transcriptomic data. So I'm looking for searches, right? These are all searches here on the left. But I have no idea where the transcriptomic experiments are unless I start opening all of these little icons here as little categories and menus. But we have this cool feature here that allows you to filter the searches. And I'm interested in transcriptomics. So as I start typing tran, you'll notice here, oh, here's a category called transcriptomics. And it includes, in VectorBase, it includes two subcategories, microarray evidence and RNA sequence evidence. Each of these will take you to a set of searches that allow you to interrogate underlying data. So I will start with an RNA sequence evidence, and I will click on RNA sequence evidence here. And this will load a list of experiments or data sets that we have loaded in VectorBase from various organisms. And you'll see this page is it's a table divided up into 3 columns. The first column is the organism that the data is mapped to. And you can see here, there's a bunch of 80s. And then as we go down, you'll see anopheles and so forth. The central portion, or the middle column, is the name of the experiment. This includes a little question mark here, which is very useful. You can mouse over it without clicking, and you will notice here that you get a summary and also a link to the publication. So this should help you identify a specific paper that you may want to interrogate, or data from a specific paper that you would like to interrogate. Finally, the column on the right here includes the types of searches that are available for this particular experiment. And those searches are described here at the top. You will notice that for the first search, for example, in the first row, there's an FC search and a P search. And what do these stand for? We'll look up here. FC stands for fold change. So you can look for genes that are differentially expressed based on fold change. And the second thing is a percentile search. And so again, you look up here, P stands for percentile. And essentially, it's you order all the genes based on their expression value, and you percent rank them, basically. So the 100th percentile is the highest expressed gene, basically. Okay, so for RNA sequence data, we take all of the raw RNA sequence data, so the FASTQ files that come from the sequence read archive that has been deposited by the people who publish the papers. Obviously, we're not generating any of this data. This data is generated by people like you. We take the data, however, and we process it all using the same analysis pipeline. So all the data, all the FASTQ files, all the reads that are coming from your sequencing runs are aligned using the same aligner, and the values or counts associated with each gene are calculated using the same software. And finally, differential expression is determined also using the same software. So this allows you then to have some confidence when you compare between experiments. Obviously, there's experimental variation depending on read count and so forth. But ultimately, if the analysis pipeline is the same, that gives you some confidence that at least the analysis is the same across all of these experiments. So you can ask questions not only within one experiment, but across experiments. So as I showed you here, this list is really long, right? So if you're looking for something specific, it's very hard for you to just go down and one by one look at these. So that becomes hard. So we did provide a filter at the top here, which allows you to quickly find experiments that may help you answer questions that you're interested in. For example, I'm going to look for an experiment that includes the word Zika in it, and I'll notice here that there are three experiments or 3 studies that come up that have the word Zika in the name, right? So in this case, it's a Zika and dengue infection experiment. Here's another one, response to Zika infection, and here's another one again with the word Zika in the name. So in my case, I'm interested in determining differences between Zika infected versus dengue infected cells, and or the differential expression between them. So I'm going to start with this first experiment up here, and I'm going to start with a fold change experiment. And let's go ahead and do that. So once I clicked on fold change right here, you'll notice now that my experiment is still listed up here. I can look at all of the experiments that are available if I click on show all, but below here, you will notice that the fold change experiment has loaded and is available to me. So now I can configure the search, which looks super complicated, but in fact, it's actually very, very straightforward. So this is, we try to order this in a way that's human readable. So for the experiment, Zika and dengue infection, this is the sense strand. So if it's strand specific, we map both the sense and the antisense, and that allows you then to look at potential antisense transcriptions, and that's obviously could be potentially very useful. I'm going to return all the protein coding genes. If I click on this, you'll notice I can select all. So this would return things like pseudogenes if I'm interested in looking at transcripts from pseudogenes or other entities that do not code for proteins. Okay, so for the experiment, I'm going to return protein coding genes, and I'm going to specifically return protein coding genes for genes that are up or down regulated. I can change this to either up regulated or down regulated, or leave it at up or down if I'm looking at just differential expression without caring about what whether it's up or down. I just want differential expression. Okay, and I'm gonna return them for where there's a fold change between the samples of 2. That's our default. I can change this, and I can change it now, or I can change it later on. I usually leave it at 2 to begin with, because that just gives me what the default value is and the results, and then I can go back and modify it if I think I want to make it more stringent or less stringent, and that really depends on the experiment. And as I go down here, there's some defaults selected, right? And you'll notice here that it's you know, in my case, I'm going to compare, for example, let's start off with Zika compared to non-infected, right? So I'm going to compare the Zika infected to the non-infected sample. And whether I select, in this case, average, well, actually you only have average as a choice, but that really doesn't matter because I'm comparing a single sample to a single sample. So you're not averaging anything in this case. If I selected more than one, so if I, for example, compared both the Zika and the dengue infected samples to the non-infected, now I can change, let's see here, one of these should change. You'll notice here that so I'm not sure why this is not changing. It's supposed to change and give me additional options, so maybe I have to reload the page. But the bottom line is I could change this to, I see, I know why, my mistake. So I'm selecting up or down regulated, so again, it doesn't really matter. But if I change this, let's say, to up regulated, you'll notice now I can change this option here to, and there are different options that appear. For example, I can do minimum in this case. So I'm saying I want genes, I'm going to return genes that are upregulated by at least 2-fold, comparing the minimal expressor from these 2 samples and minimal point compared to, in this case, it doesn't matter because it's a single sample, okay? And so this graphic here on the right really is meant to give you a visual representation of what you're selecting. So you can be confident that, oh, this is exactly what I want to find. Okay, so let's go back to just comparing Zika versus dengue, and I'm interested in up or down regulated. And in the end, at some point, you feel comfortable that, okay, this looks like the right parameters. I'm going to run the search. The nice thing about this is that if you make a mistake, it doesn't matter. It's an in silico experiment. You only wasted some time, so you can always go back and modify this. So I ran my search, I returned, and in this case, I compared Zika to non-infected, and it returned 103 genes, okay? So, and I'm gonna actually edit this and rename this, and I'm gonna call it Zika infected, because you get a default name that's provided by, you know, computationally generated, and so it may not be the best name. So now you'll notice this step is called Zika infected. But what if I wanted to find genes that are specifically differentially regulated in Zika infected cells that are not differentially regulated in dengue infected cells, or are in common between Zika infected and dengue infected cells? So then this is another huge strength of this database, is the ability to build on your searches in what we call a search strategy. So I can add a step here and expand my search. So let's go ahead and do this. So I'm adding a search. I'm going to combine with other genes. I can modify this operator here, how I want to combine the results from both searches. And I can do this now, or I can do it later. For now, I'm just going to say, well, I want to intersect. I want to find things that are differentially regulated in my Zika infected cells, and also differentially regulated in the dengue infected cells. So let's go down here and select the search. So I'm going to search for transcriptomic again, just like I did before on the main page. I'm going to select RNA sequence. This will load the entire searches. Again, I'm looking for the experiment that I used. So here's our Zika and dengue infected cells. I'll select the fold change experiment again. And I'm just going through and doing the same thing, except for now, I'm going to compare dengue infected to non-infected, and I'm leaving everything else at a default. So I'm looking for differentially regulated by at least twofold in dengue infected cells compared to non-infected cells. And remember, I'm intersecting these results with my previous results, which are the Zika infected cells. So now when you run this, this will generate a two-step search strategy, which is intersecting the genes that were differentially regulated in Zika infected cells with the genes that are differentially regulated in dengue infected cells. Let's change the name here. So I remember this because this is like a lab book. You can, if you log in and create an account, you can save this, and then you can, it's like your lab book of an in silico experiment, which you can come back. You don't have to, every time you come back, rerun the search. So here I have my 63 genes that are in common between dengue infected and Zika infected cells, and I can go down and look at them. I can change this operator and say, well, which ones are specific to Zika infected cells? So I can do 1 minus 2. So here's 1, here's 2. So 1 minus 2 would give me the ones that are in 1 in Zika. And now if I revise this step, you'll see that the result will be the genes that are only differentially regulated in Zika infected cells, but not differentially regulated in dengue infected cells based on the parameters that I used. And so there are 40 genes, and I can inspect these. And also I can modify this and change it to 2 minus 1 if I want to see the ones that are differentially regulated in dengue infected cells, but not differentially regulated in Zika infected cells. You'll notice down here that a number of these genes that are at the top here are listed as unspecific product. And so obviously that's not very informative, unfortunately, but you can add columns that may help you or may shed some light on the function of these genes. So in this case, let's see what happens if I add the column for the domains. So if I look for interpro domains, for example, you'll notice here that there's an option to add a column for interpro descriptions. So these are functional domains that have been mapped to the genes. All of the genomes in VectorBase get run through interpro scan, and we identify PFAM domains, smart domains, interpro domains, and so forth, and we map them. And then they're available to you, and you can get some information from them. And you'll notice here this column that I just loaded, that I'm going to move here to the left a little bit. You'll notice here that this product, that's unspecific product, it's an IPP transferase potentially, or it has a domain for that. There's some cysteine, whatnot. I have no idea what this does, but clearly it gives you some information about what this potentially does, and so forth. And so you have the ability to now explore this data in a bit more functional way, and you have genes that are potentially of interest. Okay, I'm going to quickly jump to microarray searches and show you one of those searches, and then I'll switch it over to Eve. So rather than going back to the home page, right, and looking for the microarray searches, notice here that in the column at the top here in the menu, there is a menu for searches, and it shows you all the categories that are available to you. And I can go in here and now search either for transcriptomic, and this should filter the results just like we did on the home page, and I can select microarray, or I could have typed microarray directly. This will take me to the microarray experiments. Now I can, same thing, you'll notice here there are different searches. Obviously microarrays are not RNA sequence, so we're not looking at reads, but rather we're looking at cDNA hybridization assays, where we're looking at color differences between, or intensity of color between samples. And depending on the type of microarray, you may have different types of searches available to you. I'm going to quickly look for an experiment that has that did something with malaria, and in my case, let's see which one. I think I will search this data set here from Pinto et al. And rather than doing a full change, I'm going to do a percentile search. So you'll notice here, these are the samples from the experiment. In this case, they used an infectious P. Bergii versus a non-infectious one. And so I want to find genes that are expressed in the higher, in the 80 to 100% percentile of the infectious P. Bergii sample. I can change these numbers, obviously, but for now, I'll keep it at default to save some time. And you notice my previous search is still up here. You know, I didn't close it or delete it, so it's still available. But now from my previous one, I'll notice here that there are about 2,164 genes that are expressed in the 80th to 100th percentile for the infectious version of P. burgii. Remember, this doesn't mean it is not expressed in the non-infectious version. It just means that it is highly expressed in the infectious version. So now what I can do is I can add another step. And once again, I'm going to go to the microarray experiment. Let's select the malaria one. I can, in theory, search Pinto, and I'll find there. In fact, we have another experiment from Pinto et al. But in this case, we're going to select the same one. I'm going to select the percentile. Now I'm going to select the one with the sample from the non-infectious version of P. burgii. And let's make this a bit wider. So anything, I want to basically remove anything that is expressed between 60th and 100th percentile in the basically the invasion deficient version of P. burgii sample. And rather than doing an intersect, obviously, I'm going to do a minus. So I'm going to basically, I want things that are highly expressed in my previous search, and I want to remove the ones that are also highly expressed between 60th and 100th percentile in my second sample. And so as we run this, we'll see what the results get back. We come back with 8 genes that meet these criteria, okay? And so now we can look at this again. Again, you have a number of unspecified product, but this interpro domain description ends up being super helpful for you, so you can actually take a look at them, and that could help you with your analysis. Just one thing to note before I turn it over to Gloria and potentially answer some questions if there are any. You don't necessarily, if you're asking a question in the microarray space, you don't necessarily have to be stuck in microarrays, or vice versa. If you're looking at RNA sequence data, you could also leverage experiments that are done with a different assay. So for example, up here, I looked for genes that are differentially regulated in Zika infected samples by RNA sequence. Then I subtracted these results from the sample with dengue infected samples, basically. So essentially, I'm asking, show me the genes that are differentially regulated in dengue infected cells, but not in Zika infected cells. I'm going to add another step just for the sake of doing this. And now, instead of doing a RNAseq search, I'm going to do a microarray search, and I'm going to see if there are any dengue experiments in here. And it looks like we do have some. And again, it doesn't matter which one we end up selecting here, but I will, let's just pick one of them. Let's pick this last one here maybe and see. I'll pick a percentile search. And again, this is just to show you that you can do this. So I'm going to say, find me anything that is, right, these are, actually, I'm going to switch to the fold change rather. This looks like an interesting experiment. So I'm going to select anything that is was expressed in the sensitive strain. So these are ones that can be infected with dengue, as opposed to the resistant ones up here. Actually, I'm going to select them this way. So I'll do this here. I'm going to select uninfected up here. I'm going to compare the infected strains and the sensitive one to the uninfected. I'm going to ask for anything that is upregulated in this stage. And we can change this to the average here. And I'm just going to run this search. And again, the result may be 0, right? But the nice thing about this is that you're not wasting any reagents. You're doing an in silico experiment. You can look at this, and sure enough, my result was 0. So none of the ones that are expressed in this, that are upregulated in this dengue experiment are the same as the ones here. And so this may indicate something interesting about this, or it may indicate that maybe you need to modify your parameters a little bit. And so I can go back here and modify this. Maybe I should just select up or down regulated and not care about the upregulation and see what happens here. And you can, and as you can see, you can keep playing around with this and see what you get. So now when I change it to differentially regulated, I get 2 genes. So there are 2 genes that are differentially regulated in the dengue infected samples from this microarray experiment, which was probably done like 10 years ago, compared, and that are also highly expressed in the dengue infected cells and are not highly expressed in the Zika infected cells, right? And so I can go down and now look at those 2 and explore them further. Okay, so I will stop here, Gloria, unless there was something else you wanted me to specifically hit on, or if there are any questions I should answer before we switch over to you.
Omar, do you just want to mention that you could add the 2 lists together because they're different studies?
So what do you mean? Sorry.
Maybe you want the results from both of those. You can just keep adding the results
here,
for more
adding. Yes, you mean adding more steps here? Is that what I mean? Or?
No, adding the results for more gene lists of interest. Step union of step 2 and 3.
Oh, I see. Yes, you're. Oh, very good point. Yeah. So Sam, what? Hopefully I understood you correctly. But you know what's cool here too is that I'm doing an intersect here, right, which is very stringent, right? So it's saying that it really must meet these criteria in both of these experiments. But it's also possible that because this is a microarray and this is an RNA-seq, that really their dynamic range is so different that it's very hard to find the right differential expression fold change or range that will actually get you many genes. So one thing I can do is I can do a union here instead of an intersect, right? And now what will happen is that it'll give me both, you know, an unredundant list basically of genes that are in this list of 50 and the genes that are in the list of 142, right? And so I end up with 190, which makes sense because the intersect was 2 genes. And so there are 190 genes that seem to be potentially important during dengue infection coming from 2 different experiments. Yeah, that was a very good point, Sam. Thank you. Any other questions or anything else before we move on? Take a look. So there are lots of questions going on. So is it possible to combine both strategies at the same time? So what you can do, if I understood the question correctly, you can run multiple strategies. And let's say I wanted to combine the results from this one. Let's say this strategy here and this one are related somehow, and I want to see if there's anything in common between these 2. I can do that. I can add a step. And now in my add step panel, you'll notice here you have an option of clicking on existing strategy. And you'll see here the different strategies that are open. I can pick, if you give it a name, obviously it makes it easier to do this. But now I select they're all called unnamed strategy, but I can give them a name, especially, you know, that makes sense. And you'll notice here I just took the results from here and intersected them here. Not surprisingly, the answer was 0, but you can see how you can build many strategies and then join between them. I can save any of these strategies, right? Call it a name, so webinar search 1 or something like that, right? And if it turns out that this is something that I would like to share with you because you know, we're working together or collaborating, I can save this. And you'll notice here I have all my saved searches here. I'll just save this with the same name. And now this will allow me to do, it will allow me to now share this with anybody. So I can take this link right here, and this will be a stable link which I will put in the chat. And anybody who clicks on this will be able to open the same exact strategy with the 0 result, right? So it's exactly the same thing that you're looking at here. And then you can modify it. You can say, oh, okay, I'm just going to go ahead and delete this last step because the result was 0, right? It won't modify mine. It'll only modify the one that's in your workspace. Okay, let's see. Are there other questions?
We're getting a lot of questions that are related to, I have 1 gene of interest and I want to see the transcriptomics for that 1 gene.
So that, I think Gloria will cover that. And so
Yes.
Wonderful.
Any other questions that I should answer before switching over? Somehow I lost the question panel, so until I close this window. So
I think we can continue, Omar. So
perfect. I will make you the presenter.
Thank you.
And I'll answer questions offline.
Okay, so are you looking at my screen now?
Yes, we can see it.
Great, thank you, Omar. Okay, so now again here in the VectorBase website, so something that Omar was showing you is the data sets that we have. So how do you know which data sets you can use for all that nice strategy that Omar was showing you? So you come to the homepage and you click on data and select data sets in VectorBase. So notice we have 483 data sets, and this list increases every 2 months. Actually, today we have our release, and the data sets and the website will change. But from all these data sets, you want to know which ones we have from RNAseq. So you click here and say from category, which is this column here, and you type your keyword. So I only want to look at the RNAseq. So it's showing me that we have 327 RNAseq data sets in VectorBase. And you can do the same for any other, use any other keyword to know what we have. You can repeat the same thing again, data sets in VectorBase, but change the category for microarrays. So it's showing you we have 49 microarrays. And these are the data sets that you have available in VectorBase to do your analysis. So how do we obtain these data sets? We curate the literature. If we do not get fast enough to a data set of your interest, please let us know. Data from publications, you just email us. But if it's your own data, the one that you produce in your lab, all you need to do is let us know what is the INSDC accession number you could have deposited in ENA or SRA, and from there we export the data to have it available in VectorBase. So if you want to be able to say, oh, in your paper, my data is available in VectorBase for search strategies, you need to contact us as ahead of submitting to the journal as possible, because it takes time for us to load the data. We have a waiting list, so we appreciate as much time in advance as possible. Okay, now and to which genomes are these microarrays and DNA and RNA-seq experiments aligned to? These are the genomes that we have in VectorBase, and to these organisms is to which we align data. And that's data, organisms, and these are the 46 genomes, depending, if you do on Aedes aegypti RNA-seq experiments, well, that's to which we will align it, of course. Okay. Now, I want to explore, and this is answering the question that Sam said was in the comments. I want to explore transcriptomics for a specific gene that I am interested. So you come to VectorBase, site search, and you can type here a gene ID or a keyword. Because I type it a gene ID, so site search is giving me this card here that says this is your exact match, and that's my top hit. So I click on this and it takes me to the gene page. So what is a gene page? It's like a CV, is the curriculum vitae of a gene. Here you will find in silico, in vitro, and in vivo data related to this gene of interest. So here at the top of the page, you find the gene ID, and here is the product description along with a lot more information. Here we have the shortcuts and the table of contents. Again, the same way that Omar showed you, you just start typing words and autocomplete will tell you what is the place that most likely you want to go. And notice that what we want is transcriptomics, and we have it here, and also we have it here in number 8. So if I click in transcriptomics, I will land in section number 8, and this has 3 subsections. So we have the RNA-seq transcription summary, and these one are all the experiments that provide experimental evidence for this gene using only RNA-seq data. The other one is transcript expression, and this combines both the experimental evidence for microarrays and RNA-seq. And the last one is when we are scheduling the data sets, if sometimes we could upload the user transcriptome graphics. Okay, so let's go back to the first one, RNA-seq transcriptomics summary. Oh, sorry. Okay. So here in the x-axis is the level of expression, which you can change to either log scale or linear scale. And in either case, log or linear scale, we are showing the things in TPM, which is transcripts per million. And if you click here in data sets, it will bring you to this page, and it's telling you that for this particular gene, out of all the RNA-seq experiments in VectorBase, 13 align to this gene of interest that you are looking at. Okay, going back here, if you click here, read more, you are going to have these bullet points in which we hope help you understand more this graph. And here, notice if I hover, I have this menu here, so you can zoom in and out and see the labels, take a screenshot of what you're looking at. Below this graph, we have here the transcript expression, which I said is combination of microarrays and RNA-seq. Again, the same thing, click on data sets and you will have a list of all of them. What are the columns that I have? So I have preview, name, summary, attribution, assay, and notice that all these columns have these arrows, which means you can sort them. And the last one, assay type, means that you can have either microarrays, or you also have RNA-seq here available. Okay, let's look at the first one. Here on the x-axis, you're going to be looking at the different samples, and here the level of expression, or the sampling points could be as well for the particular experiment. On the side, you are going to have a data with exactly the value that we are plotting here. And if you were looking at, for example, if you click here in the attribution, this is the description that we provide for you. We wrote this in VectorBase when we curated the data. But if you want to know exactly what the author said, you click here in attribution, and you come to the PubMed page. Another thing that you can do, and something that is different, that is not available in the microarrays, but is available in the RNA-seq experiments. So, for example, this is an RNA-seq. In addition to the graph, we are also showing you coverage plots in the genome browser. Okay, so once this loads, okay, there you go. So what happened here is this is my gene of interest. And the reason why I have so many lines is because this gene has many alternative splice variants. So my gene is AGAP004707. And I have the same gene ID with RA, RB, C, D, E, F, because these are all the different splice variants, and notice how they differentiate in either the 5' or 3' area, or they even have different exons along the way. What should have happened here is that we had a track with the coverage plot, which is not showing, but I have that covered. So click here and view in genome browser. And this is what you should have had. Well, we're gonna get this fixed. Sorry about this. Okay, so you come here, and here, this is the genome browser, and this is showing you for this gene that I'm looking at, how much I have evidence for each one of these exons with the RNA-seq data that I have. So for example, the experiment that I have selected here, notice is deltametrin tested in 2 samples, susceptible, here I have my resistant mosquitoes, and here I have my susceptible mosquitoes. And I have 2 tracks. Well, for now, I have only selected unique tracks, but I could select also non-unique tracks. I'm going to show that in a minute. And here, something interesting to notice, maybe you are looking at, well, but how come the gene is finishing here and I have more expression going to this side. So this could mean either that the UTR region has not been annotated, or that we are even missing a complete gene that should be there. And to fix things like this, when you're looking at RNA-seq, we encourage you to go to Tools, Apollo, and correct that manual annotation, because there is something going on here that is missing in this track of the annotated transcripts. Something else that you can have here, and if I move to the side, for example, I can move anywhere in the genome where I want. Notice if I move to the side, this coincides with this gene, the expression here, and these RNA-seq reads coincide with these genes, and these coincide really good there. Another type of track that I can have in the genome browser is that, okay, I am not much interested in look at the data by divided. Here I have the resistant and the susceptible tracks. I just want to see all for my experiment of interest, for the deltametrin treated mosquitoes combined. So that's when you use, instead of using a coverage track, you use a density track. And here the gray is just showing you the saturation of color is indicating higher expression, independent of which sample it is. Something else that you can do is, I here open a different example. So not looking at the deltametrin experiment, the track is showing up in orange. But I open another experiment here, looking at Plasmodium bergei infection in Anopheles, that's the species that I'm looking at. And as Omar mentioned to you when he was doing the searches, sometimes the data is stranded, and if it is, we color it. So for the genes that are forward, which in the genome browser appear in blue, the RNA-seq track evidence is also going to show up in blue. And for a reverse gene that shows up in red, the RNA-seq track evidence is shown in red. And notice how it really coincides really good, the exons with the experimental data that we have. And if for some reason you think, well, look, there is some gaps here, or maybe we are missing some splice variants, and maybe in one of the variants, this is actually divided in many different exons. But how can you do all this custom of adding and selecting different tracks? You click here on Select Tracks. If you click on Select Tracks, this is what you are going to see. And if you're interested in transcriptomics, so you click here, we have available for you 848 tracks. You select here, these are all the different experiments that we have available for this species. We are looking at Anopheles gambiae. And you can here select, I want the coverage plots that are the ones that I was showing you in colors, or you want the density ones, the ones I was showing you in gray. And I was showing you only the unique tracks because I wanted to know if you have a gene family. So some reads can go to multiple places in the genome. So sometimes you want to see that, sometimes you don't. I was showing you only the unique tracks. And what I was showing you as red and blue is because the data is stranded. So here you select, you want to see the forward strand or you want to the reverse strand. And some data is not stranded. For example, the deltametrin one, so not stranded data, and that's showing up in orange. Something else that we have here for you is that here we are looking at, you hover with the mouse and notice we have this arrowhead here. You click on it, and right now I'm showing you the data in log scale, but if you want to see the data in linear scale, you can unselect that. And you can also, change the height of how you're looking at the data. We combine all the data, and if you, select here the track, you can know more details about the tracks. We have combined all the RNA-seq data and said for all the RNA-seq evidence that we have in VectorBase combined, this track is providing you with those introns that are supported for the evidence that is supporting the introns that exist in the genome. So for example, notice this one here, this is a thick line compared to these ones here. So we have more evidence for these thick line introns that, let's say, for these introns right here. And notice how this intron coincide with this intron that we have here, and this one here, and sometimes it doesn't. So it could mean either the annotation is wrong or that we are missing a splice variant. I think that's the same. Oh, something also that is very important is that, okay, you hover with the mouse and those numbers that are here are, you know, the transcripts that you have per million. They are the support to give you the of how is represented the abundance of those RNA-seq reads when aligned to this genomic region. Why this is not constant? Why we have peaks that go up and down? Again, same thing. Probably is, you know, different splice variants, or these are exons, and that's why the data is not consistent, just starting and going straight. That could be something. And I think Omar or Sam, I don't know if is there any other question or something that I didn't address from the questions?
There are multiple questions. So
Good.
there are a couple that are potentially worth demonstrating. And so there was a question about nested strategies. There are a couple of questions about orthology, and there was a question about gene families. You addressed a little bit about the gene families where you said that we have unique and non-unique aligners available.
Yeah.
But I'm happy to address it in a strategy context if that's useful for people. And then the last question was how can I get a certificate? And automatically you'll get a certificate tomorrow by email if you need one. I know for some people you need to report this for taking time off, and so that certificate will come to you tomorrow automatically. So Gloria, I'm happy to switch over and cover a couple of other things.
Sure. Yes. Okay. Let me make you the presenter.
Okay, so everybody can see my screen. While we were talking, I added another search here. I just searched for serpin genes, which are a gene family Anopheles. I'll come to that in a second, but I just wanted to search that includes serpins in here. So the first thing I'm going to address is there were two questions actually that relate to leveraging orthology. One was, can I combine searches from an experiment in one species with searches in an experiment from another species? And the answer is yes. You have to convert your results from one species to another one using the orthology tool, and I'll demonstrate this. There was even another question where somebody asked, well, can I actually ask a question about even another species, another organism that's in ViewPathDB, not a vector, right? So let's say you want to compare between genes, for whatever reason, genes that are upregulated in, well, there could be some good reasons, but let's say genes that are upregulated in Anopheles during infection, that are also upregulated in human macrophages during, a malaria infection. Let's say they're both Plasmodium infections. Or not macrophages, obviously with Plasmodium, but you can pick a related organism. And so let's say Toxoplasma. And so so let's say you're comparing, you have a malaria infected experiment in mosquitoes, and you want to know if some of the genes in the vectors have orthologs in macrophages that are infected with Toxoplasma, for example. And the answer is yes, you can do that. What you do is you have to go to viewpathdb.org, so the main portal site, which includes access to all the data. And you can run the same types of searches there where you use then leveraging orthology. So you'll transform your Anopheles genes to human genes, or the human genes to Anopheles, depending on how you did this. So in VectorBase, I can quickly go in here and say this experiment was done in Aedes aegypti in this case. But let's say I'm looking at these 190 genes, and I'm curious if Anopheles has orthologs of these genes, and maybe I want to add experiments from Anopheles. So I can add a step here, and rather than combining with other genes, I can transform to related records. And you'll see here there are different options. In my case, I'm interested in orthologs. So I can select orthologs. And now I can filter this and search for, in this case, I want to look for Anopheles gambiae pest. So I'm just typing pest to quickly get me there. And before I click on run step, I want to say that there was another question about whether I can find the orthologs in Drosophila from Nelson Lau, and the answer is you will later tonight. So we are releasing today, and that'll include Drosophila genome, and it'll allow you to easily then also select Drosophila from the list here, and then click on run step. And then what will happen is you will get a list of orthologs of your 190 genes that, but rather than now looking at ADs, we're looking at Anopheles. I can go down here and look at them. So these are my Anopheles genes, and you can see here the ADs ortholog and the orthology group that they belong to. And you can see here a bunch of these belong to the same orthology group, so maybe some kind of a gene family, or they're all cytochromes. Okay, so, and then I can, now I'm in Anopheles genes, so now I can add Anopheles experiments if I want, okay? So there was another question about, well, what is the nested strategy? Why would I want to add a nested strategy? And the idea of a nested strategy is that you see here, the strategy here is composed of steps. We have 1, 2, 3, and then 4. Now let me go ahead and add a step and ask how many of these genes are potentially secreted. So how many of them have a potential secretory signal peptide? Again, I don't know the answer. In this case, we're looking in Anopheles. So I'll go ahead and run the search. We'll see what happens. And so there were 83 genes out of these that also have a predicted secretory signal peptide. But let's say I want to know how many of them, how many of these 320 also have a transmembrane domain, or how many of them have both a signal peptide and a transmembrane domain. So if I add a step here, I'm actually limiting on these 83 genes, not on these 320. And so what I can do is I can take this step here and make it into a nested strategy. And what happens, it's going to take this step and put it out in its own little mini strategy where I can add additional steps. So now I can say how many of these I want to add transmembrane domain search. And in my case, I'm going to select a union because I'm looking for anything that has one or the other or both, basically. And again, we're in the Anopheles world. And I'll go ahead and run this. I can change the number of transmembrane domains if I want, but let's leave it at defaults. And now what you will see is this search here of union between signal peptide and transmembrane domains gives me 5,000 genes or about that. And now I'm feeding the results of this nested strategy into my main strategy. So now there are 206 of my orthologs of all of these steps here that have either a signal peptide, a transmembrane domain, or both. And so quickly then I'm gonna go down here. I know we're beyond the hour, but I think this is probably very useful. And since it's recorded, people who missed this can always come back and look at this. These 21 genes down here, there was a question, how can I look at multiple genes or gene families? So obviously, depending on how the aligner is working, you know, they may not align, and how to calculate expression values for each gene, obviously, so they all work in different ways. But we apply the same algorithm on all of our sets. And so here I'm looking at 21 genes from Anopheles. So you can upload your own list of genes. There was a question about that. So I can, there's a search here for gene ID. So I can go and if I do ID, there's a gene ID search. I won't go here, but we can always send the link here. But you can upload your own list of gene IDs. So instead of doing a search, you just upload your list of IDs. They'll show up here. And now I can either ask questions about them from an experiment and say intersect this with, you know, the P. Bergii infected experiment, or I can add columns here on the to my list of genes that includes the graphs from the various experiments. And so I would have to go in and look for one of the Anopheles experiments and look for, there's a lot of them who have all the expression values and so forth. And then let's say I can pick, again, I don't know what this experiment is, so it may not show us anything. I'm just selecting a couple of columns quickly. And if I add them, it will load expression graphs here. And so now I can quickly look and see for my set of serpent genes, I can go down and look in this experiment and see what the graph looks like for each of them. And then maybe pick from those to look at them in a bit more detail. Okay, so I think this is everything I was going to show quickly. I know there are many more questions coming in. Gloria, what would you like to do? I think we're probably towards the end. So.
Let's do this. I'm just going to wrap for the people that wants to leave, but let's stay to answer all the questions because that's the whole purpose of our webinar is to have, you know, a live conversation. So for the ones of you that have to leave now, thank you very much for attending today's webinar. And as I said, we're going to stay answering the questions for the people that still have questions. See you next week for a release. As Omar said, we are releasing today. So in next week, the webinar will be about what we release, what are the new data sets that we have. And in 3 weeks, we will have the next and final webinar of this mini series. And in that one, we will use Galaxy and other tools in VectorBase to analyze your own data. For example, you generate data in your lab and you want to do your own differential expression analysis, or you want to call your SNPs. Those are the kind of pipelines we're going to run next week. So see you if you have to leave now. And Omar, you can continue answering the other questions if there are more.
Somebody's asking about, will there be a copy of all the questions and answers available? Gloria, we could, in theory, look at all the questions and filter them and then send an email to everybody with sort of the answers that we provided.
Yes, yes, we could do that. Something else that we are working on is some FAQs, and I see that some of those questions are being worked already by Sarah and Sam, the FAQs.
Right, right. So there was a question about single nuclear polymorphisms. And so we do include VCF files in our genome browser. And so you can turn on VCF files and look for a specific gene, look at the polymorphisms. We are working, so for other ViewPathDB sites, we've loaded and recalculated variants, and then we provide searches against them. So you could say, well, find me all the genes that have a certain number of non-synonymous SNPs when I compare isolates that come from West Africa and compare them to isolates that come from East Africa, for example. And so you can imagine doing something similar for vectors. We don't have those queries implemented. We need to develop and update our pipelines. But the VCF files for many experiments are available in the genome browser. And just as Gloria was showing you how to load tracks, you can just load the VCF files and look at SNPs that are specifically in your genes of interest. There was a long question about annotation and RNA sequence data, and sort of questions about Apollo. I think I will leave this one maybe to answer offline, Gloria. It was there are multiple things in there. So
I found it from Bila, which is a great annotator. She has made a lot of annotations for the Glossina genomes. So yes, I will come back to you, Bila.
So then there is, let me see if we're getting, and I apologize if we missed any of your questions. I'm sure we will be able to look at them afterwards and make sure we follow up with you.
On my screen.
Then the other question about the genome browser, is that results in histogram just one experiment or a combination of multiple experiments in paired end reads? So yes, I mean the paired ends are the histogram represents the paired ends, but of course if it's strand specific, and each sample from the experiment will be a separate track. Okay, so obviously if it's paired end or single end, they'll be represented in a single track, but obviously different samples are on separate tracks, and then unique and non-unique are on separate tracks as well. So the reads that map uniquely to a region show up in the non-unique tracks.
And Omar, something that I forgot to mention is that once you select what you want to represent and you click here, or with a keyword you type introns or anything that you want to select, you need to click on back to browser to load them here.
Yes.
Big thing that I forgot.
Yeah. Okay, I think we're, I'm sure there'll be more questions. As Sam indicated in the chat, feel free to click on the Contact Us link in VectorBase, and we will be more than happy to answer your questions by email. The webinar is recorded, so you'll get a link to it tomorrow, and also we'll post it on our webinar page. Gloria, anything else you want to add before we close?
No, thank you very much. It's been really good. People asking questions, that's what we like.
Absolutely. Thank you very much. Okay, bye everybody.
Bye.